Hi everyone, my name is Kapil Devkota, and in this talk I'm going to present a new way of predicting PPI through sequences, which also uses some network theoretic principles, a method called Topsy-Turvy. Now to describe the basic premise of our work, we are given two proteins with known polypeptide sequences. Because of the genomic sequencing revolution, sequence data for proteins is often readily available, even when structured data is not. Now we can visualize a model that somehow can take these sequences and predict if the two proteins actually bind or not. So the binding is going to basically be a probability between 0 and 1. And as the probability becomes closer to 1, the model becomes more confident that the two proteins will actually bind. I would like to stress the fact that we are actually not trying to figure out how proteins interacts or the mechanisms of protein interacts. Like the methods highlighted in the top left portion of this chart, what we are actually trying to develop is a high throughput model that can predict whether or not the protein actually interact, which is basically a binary problem. That is why our model is at the bottom right portion of this chart. So this is the basic diagrammatic representation of the sequence-based model for predicting PPI. We are given two sequences. The embedding model basically changes the proteins into points in space from which we can do machine learning to get interaction probabilities described here. So there is actually a lot of work done on this kind of modeling. For example, in Recom 2021, we presented a method called Descript, which is exactly the same kind of sequence-based structure. And the machine learning part is a deep convolutional structure shown here in the middle. Additionally, it also has a contact map layer at the end for interpretability purpose. So even though Descript does not take structure as input, the contact map is designed for the model to be structurally aware for the mechanisms of interaction. Finally, to produce a genome level network, what you need to do is choose a proper threshold for calling the prediction true or not. And as we are looking at the entire genome, we have to cover hundreds of millions of protein pairs. So the prediction phase has to be really fast. I'd like to highlight this point. Now, while we are talking about this sequence-based model for predicting PPI, there is an entirely different way of algorithms that can parallelly do the same task. In fact, before I like to draw an attention to an entirely distinct way of predicting PPI, the one that uses existing network for predicting new interactions. These methods, broadly called network-based models, are in fact very well studied and very diverse, and they range from using small local neighborhoods to predict interaction, for example, the triadic closure method that we describe here, to the methods that span the whole network that convert the node in the networks to points in space from which we can do downstream analysis. There is also a new method called Glide, which we presented in ISMB 2020, one that combines both this topology-based and neighborhood-based approaches. We especially found them to be very effective when the network data is available. So this network-based approach, which we also call top-down approach of predicting PPI, is very distinct from sequence-based model like Descript, which uses sequence information only. So the major objective of our talk is, can we combine these two approaches? In other words, make the sequence-based method that is aware about the network at all. Enter Topsy-Turvy. This is our new model, one that incorporates both top-down network view in the training phases, but for prediction phase, however, we use sequence only, like Descript. Now this table shows the type of data different PPI prediction algorithm use, both while training and while predicting. We see here that while Descript, which only uses sequence, and Glide only uses network, Topsy-Turvy uses both sequence plus network during the training phase and sequence only during the prediction phase of the algorithm. So what does this model look like? Here we describe the diagrammatic sketch of Topsy-Turvy. The top part here is the network-based view. This is passed to the Descript model during the training phase, and the sequence is used for predicting PPI relationships. Now, how are we going to train this model? So we basically use stochastic gradient descent. This is the basic training diagrammatic sketch of the training phase of the model. Here we see three major losses: glide loss, BCE loss, and regularization loss. The first loss term is a BCE loss, which says if the prediction is actually accurate or not. We have label data and we just compare it against the label data and predictions. The second loss here is the regularization loss obtained from intermediate contact map that we described earlier. This is motivated by a similar loss function in the script, and it helps with interpretability. And the final loss is the network derived loss from the glide metric, which basically says whether or not the proteins are actually network related or not. To go deeper into this glide loss, we set a parameter called glide threshold, or GT, as we describe here. And any pair with glide similarity greater than GT is considered by the model to be a pseudo-edge. Then we apply binary cross entropy loss to see if the model prediction agrees with this synthetically created new network edge. So there is a question: why should this network-aware training procedure help in the model becoming more effective? It is because we have found that network-based approaches are standalone very powerful if the network information is sufficiently present. And since this is usually satisfied during the training phase where we are dealing with a source network of a source species, we expect the network enforced training to cause significant gains when combined with sequence data. So let's talk about the way of evaluating our models first. We trained Topsy-Turvy along with the other models on human string network and tested whether it can predict PPI from sequence alone in the following 5 organisms: mouse, worm, yeast, fly, and E. coli. We only looked at sequence data in our experiments. So here are the results. We can see here that Topsy-Turvy is outperforming other sequence-based model in all organisms. I specifically want to emphasize that we are using only sequence information from the target species, but we are able to outperform all the existing methods. So it looks like we are learning some kind of transferable PPI information from human string PPI to other organisms. Furthermore, to validate the strength of PPI prediction using Topsy-Turvy, we wanted to see if the prediction is solely due to ascertainment bias. Now, what do I mean by ascertainment bias? We know that existing ground truth of PPI data favors well-studied regions of the network, one where biologists do a lot of experimentation, which makes those regions very high degree. Since this data is fed directly to the top-down model, this model can directly see this kind of bias. And it might be unfair to compare it with sequence-based methods, which only has access to sequence information, not this biased data, network data. Since this ascertainment bias is more pronounced in regions where degrees are high, what we did was we actually devised an experiment where we compartmentalized all the network edges based on their maximum degrees, and we individually computed the AUPR scores for each compartmentalized degree ranges. We see the results in this plot. As the plot indicates, the performance gain is uniform across the network, even on low degree regions, which is here. So both high degree and low degree connections benefit from this kind of approach. And as a side note, I'd like to highlight the fact that it is interesting to see that sequence-only methods like Descript are also more effective in predicting high degree connections. As we see here, the improvement is drastic here. We believe this is because significant majority of sequence pairs that we use to train the Descript model is also from these well-studied regions. So there is the indirect bias coming from the network that also seeps in for sequence-based model like this. Now, as a brief recap, we discussed about Topsy-Turvy, which takes both sequence and network during the training phase, but only uses sequence during the prediction phase. What if we are dealing with organisms like fly, which already has a sufficiently large PPI network, PPI experimental connections, and we are trying to include it in our prediction phase as well. This figure, the organisms here is fly, and we have a sufficiently large experimental PPI information from fly, and we want to use them in our prediction phase as well. As you can see here in this figure, the fly data as significantly large number of experimentally obtained PPI information. So it would be suboptimal to construct fly PPI graph while ignoring already existing connections. To solve this, our approach, we devised a hybrid model called TT hybrid, which uses both sequence and network information during prediction phase as well, unlike Topsy-Turvy. So as this table shows, Topsy-Turvy TT hybrid, like Topsy-Turvy, uses sequence plus network during the prediction phase, but unlike Topsy-Turvy, it also uses sequence plus network during the prediction phase as well. Structurally speaking, Topsy-Turvy hybrid is basically a weighted linear combination of glide and Topsy-Turvy. And in our experiments, we saw that this hybrid model is more effective than using sequences or networks alone. But the improvement here is slight. We see Topsy-Turvy improving nonetheless, but the improvement here is slight. So why do we have to bother with this hybrid approach if the improvement is very slight? This is because the target network that we describe here is normally very sparse, and it contains isolated node pairs which have no surrounding network information. So for isolated node pairs like this, if you want to predict whether or not the two isolated node pairs interact, the only information that we have is the sequence data. So what we have to do is you have to use methods like Topsy-Turvy to resolve this kind of connections. So we need a hybrid approach which works well on comparatively dense regions and also on isolated node pairs. Lastly, we did comparisons with AlphaFold multimer. AlphaFold multimer is a sequence-based method which uses structure during the training phase. And one immediate observation that we came upon was that Topsy-Turvy was in fact very fast compared to AlphaFold multimer during the prediction phase. It was about 100,000 times faster. So highlighting this computational cost required for AlphaFold multimer, we had to look at only 18 protein pairs. We selected the protein pairs based on the ground truth data, whether or not the proteins pairs are actually true edges or not, and Topsy-Turvy prediction, whether or not Topsy-Turvy predicts it highly or the Topsy-Turvy prediction is very low or medium or something like that. So this gives us 6 combinations, and for each combination we randomly selected 3 candidate pairs. And we chose the settings because we wanted to also look at cases where Topsy-Turvy was wrong, and we wanted to know if AlphaFold multimer would correct that wrong output from Topsy-Turvy. So doing the experiment, we found that there was moderate level of agreement between AlphaFold multimer and Topsy-Turvy on this selected set. And we also found that AF multimer and Topsy-Turvy prediction were of comparatively equal quality, similar quality. So finally, in conclusion, we devised Topsy-Turvy to associate both top-down and bottom-up paradigm in its prediction. And we designed it to only require sequence data in its prediction. The Topsy-Turvy performance was found to be not entirely due to ascertainment bias. When some target PPI is available, Topsy-Turvy hybrid outperforms all other methods. And methods like Topsy-Turvy and TT hybrid are computationally efficient enough to make genome-wide PPI prediction tractable. Finally, I'd like to thank my collaborators, and I'd like to thank NIS and NSF for funding our research. Thank you.